Okay, I just want to say a quick hello and thank you for joining us today for our COVID-19 update and Q&A session. Great way to start off the long weekend if anyone has the Monday off. My name is David. I'm the Programs Manager for Immunity Canada, and I'll be your host today. And I'm joined by Fiona, who's the Project and Volunteer Coordinator, and she'll be providing background support for us today. Just very quickly, I just want to say thank you to our sponsor for today, Moderna, for helping us put on this event. So a very warm welcome to the over 70 people who've registered for the event. I'm joining you here from Victoria, our office in Victoria in British Columbia. And just want to start by saying we acknowledge the traditional and unceded territories of the Lekwungen-speaking peoples, and we recognize that the land's historical relationships with the Songhees, Esquimalt, and WSÁNEĆ peoples have existed since time immemorial. I think a lot of you know who we are, but our mission is to empower Canadians impacted by immunodeficiency disorders to live well. We're a member-based organisation working with patients and their families, health professionals and others who want to help ensure everyone has access to the resources they need to thrive. I want to say a huge thank you to our panelists today, Dr. Cowan and Dr. Ritchie, who should be joining us shortly. Dr. Cowan is an associate scientist at the Ottawa Hospital Research Institute, assistant professor at the University of Ottawa, cross-appointed assistant professor at the Department of Biochemistry, Microbiology and Immunology, and a physician for the Division of Infectious Diseases at the Ottawa Hospital. Dr. Cowan currently runs an immunodeficiency immunoglobulin treatment clinic in Ottawa. The clinic is expanding and is also served as a resource for clinical research. Dr. Bruce Ritchie is a professor of medicine and hematology at the University of Alberta in Edmonton, where he's active in both clinical and basic research. His clinical interests are many, including primary immunodeficiency, and he has developed specialty clinics in Edmonton to provide comprehensive care, including home treatment with blood products for patients. Welcome again, and thank you so much for joining us. Very quickly, after this introduction, we'll hear from Dr. Cowan about COVID-19, current COVID-19 research, and then we'll hear from Dr. Ritchie about vaccines and variants. And there'll be about 20 minutes sessions each, and then there'll be plenty of time for Q&As before we wrap up. How you can participate, at the bottom of your screen, there's a Zoom chat function and a Zoom Q&A. So any questions that you have, if you can put them into the Q&A, we'll save them up and we'll be asking the panelists at the end. And if you have any technical issues, you can join the Zoom, you can put them in the Zoom chat function and we'll try and help you out. Just to let you know that this session is being recorded. Only the staff here and the panelists' videos will be shown, but questions from the chat will be included in that recording. And the recording is just for people that registered but couldn't make it to this actual event. Very quickly, Immunity Canada, we offer an array of presentations developed by health professionals, our members and others. The views and opinions expressed by guest speakers do not necessarily reflect the views and opinions of Immunity Canada. Information provided at events should not substitute advice from your physician or other qualified health providers regarding your medical condition. And with that said, it's time for the main event. And so I'll stop sharing my screen and over to you, Dr. Cowan, if you'd like to share. That's great, thank you.
Okay, now can you hear me as well? Yes, okay, perfect. Well, good afternoon, everyone. Is it good morning there for the West Coast? Thank you for having me joining you today to provide you with updated information on the COVID-19 vaccine research. So disclosure, I received some funding from various companies for non-related topics to the talk that we're doing today. And I am a member of Ontario Immunization Advisory Committee. I'm a member of Ontario Blood Advisory Group and a member of Immunoglobulin Advisory Panel. As well, my view on the presentation today is not a representative of these organizations. The other thing that I forgot to update Dave was that I was promoted to be an associate professor not long ago. The objectives for the talk today will be to review updated data on COVID-19 vaccine responses among individuals with primary immunodeficiency or inborn error of immunity. I showed this slide last year. So the reason why we get vaccine is because we want to protect ourselves from the germ. And the response to vaccine is not, one way. There are multiple components of immune system that work in responding to the vaccination. One part of it is the B cell component. So the B cell responds to the vaccines by producing antibodies specifically to the antigen that they see. And apart from that, you also have T cell response. In majority of cases, once the T cell can recognize the antigen at hand, it produces interferon gamma. So lots of cytokines that can help with either killing the infected cells or helping other cells to be more effective in killing the viruses or the infected cells. So this was last year. We presented the information about COVID vaccine on September 11th. And this was the table I presented last time. By that time, there were 6 studies across the globe talking about vaccine responses in primary immunodeficient patients. So I'm not going to go into the detail, but just to remind you of what we talked about last year, that there were 6 studies, and across all of this study, about 80%, I would say 70 to 80% of patients with primary immunodeficiencies have some response with regards to B cell response to the COVID-19 vaccine after 2 doses. And also about 80% of people have T cell response as well. So what's new in 2023? There was a follow-up study from the group in the Netherlands, where last year they reported the vaccine response in 505 primary immunodeficient patients, which was a massive study. At that time, they reported on the responses after 2 doses. Now they have follow-up data after dose 3, and their data is up to 6 months. 4 to 8 weeks after dose 3. As in many research studies, sometimes we start really, really high, like they got 505 initially, but down when they wanted to understand the vaccine response after dose 3, only 47 or 50 patients left. So about 10% of the initial cohort. This graph shows the antibody titer against S antigen, which is the marker for us telling people if the B cell response is great. The first panel here is the healthy control, and they presented the data based on the timing, so 28 days after dose 2 and 6 months after dose 2. XLA is X-linked agammaglobulinemia. The third panel, combined immunodeficiency, CVID, common variable immunodeficiency disease, IgG or specific antibody deficiency. So sorry, IgG subclass deficiency or specific antibody deficiency. And then people with phagocyte defects and lastly, undefined immunodeficiency. So if you compare across the board with the healthy individuals, the antibody response, so just look at the first half, which is the 4 weeks after dose 2 first, okay? These bar represents the, I think it's the median, and then the error bar is the interquartile. So the median response of antibody titer in immunodeficient patients are somewhat lower than the healthy individuals, particularly the X-linked agammaglobulinemia CVAD patients. And the rest of the groups like combined immunodeficiency, the subclass deficiency, phagocyte defects, and then defined are not so much different to the healthy individuals. Then what about 6 months post dose 2? I'm not sure how applicable that is to immunodeficient patients in Canada, because I think we promoted patients to get the third dose about 3 months after the second dose. So not many people actually wait until 6 months after the second dose. But here you can see that for CVAD patients, for example, when they wait for 6 months after the after dose 2 before they went on to get the third dose, there is a moderate reduction in the antibody titer, but still majority of cases still have antibody titer above the cutoff of positivity. The X-linked agammaglobulinemia patients, as you know, they don't respond very well in terms of B-cell response, antibody response. And then in terms of neutralization, neutralization means the antibody that can actually bind the virus and neutralize it. Basically, it's just straightforward, neutralize antibody. And you see that at 6 months after dose 2, there's some reduction, but still above the cutoff of positivity. This one is the data, the T cell data response. The way of representation is very similar to the B cell response. And that 70%, 60-70% of immunodeficient patients do have a T cell response. And that declined a little bit at 6 months after dose 2. Now, as I mentioned to you, only 50 patients left for them to do analysis for response after the third dose. And actually, it was just only 5 weeks after the third dose. They divided patients into people who already had, actually they divided people into those who respond to antibodies after 2 doses and those who did not respond to antibody after 2 doses, and see if there is any increased response after the third dose. So for those people who already responded to 2 doses, had slightly increased antibody titer after the third dose. For those people who did not respond to 2 doses, majority of them still did not respond to the third dose. Whereas for the T cell response, there essentially was a reduction in the T cell response at 6 months after dose 2, and then back up again after dose 3. And this is for people who were responder before. People who did not respond before, some of them did come back up after the third dose, but some of them still quite low. But you can see that these data points are so minimal. There are only 1, 2, 3, 4, 5, 6 patients per group. So that's not a large number to make any significant conclusion. And I think a majority of our patients here are more interested in, okay, well, who gets sick, who get to the hospital, who still die from the infection. If you look at this data that was published in actually this year in Frontier Immunology, they found that in patients with primary immunodeficiency who ended up having moderate to severe COVID-19 infection, they actually have higher pre-infection immune dysregulation and disease activity score. And what that is that they already have baseline comorbidities, such as cytopenia, such as having, bronchiectasis, or being on immunosuppression, or many conditions with immune dysregulation. So if you just a plain immunodeficiency, had a history of infection in the past, but doing well on immunoglobulin, those people are very unlikely to develop moderate to severe COVID-19 infection. Usually they're either not getting COVID at all or having very mild disease. And this paper showed that people who still died after getting a booster in England, and this is general population, not particularly primary immunodeficient patients, the significant risk factor for death is actually age. The older you are, the more likelihood that you can have severe infection from COVID. And then when they looked at the demographic factors, what are the most, apart from age, what else can make people be at risk of death post-booster with Omicron? So the first one, severe combined immunodeficient patients are still at the highest risk of death post-booster, followed by patients with blood cancer, cancer of bone marrow, and some neurological condition. And then there was also a case series of COVID-19 infections requiring hospitalization in patients with primary immunodeficiency. In this case series, there were only 5 hospitalized patients, and among this, 1 person died. If you look at the detail of these 5 cases who were hospitalized, one has CVID, the other one CVID like with mutation in the NF-kappa B gene. Third is X-linked agammaglobulinemia. The fourth is the APDS, and now I cannot remember the full name, but it's dysregulated immune impairment. And then the 5th case is the CVID. The age range, 39 to 69. All of them, you can see all of them have comorbidities. The first one had rheumatoid arthritis on rituximab. The second one also had bronchiectasis, splenomegaly. The third patient had cardiomyopathy, atrial fibrillation, chronic kidney disease. The fourth one, who actually died, had non-Hodgkin lymphoma. So this person also had blood cancer as well. The 5th one has a chronic lung disease with institutional lung disease, bronchiectasis, colonization of aspergillus. The person also has Sjogren's syndrome or chronic kidney disease as well. And so the message here is just to tell you that patients who have severe COVID infection are those who also have comorbidities and at a higher age range. You may have a question, what is the effectiveness of the booster formulation against severe Omicron infection? So this data came from North Carolina. And this is general population. This is not particularly patient with primary immunodeficiency. They looked at 6.2 million residents in North Carolina and divided people into 2 groups, those who received monovalent vaccine and those who received bivalent vaccine. So the monovalent is the blue line, and the bivalent is the orange or red lines. Okay. And the graph shows effectiveness of the vaccine against hospitalization or death. Basically, at 2 to 4 weeks after getting monovalent vaccine, the effectiveness of vaccine is going up really high, up to about 60 to close to 80% in preventing hospitalization or death. However, that effectiveness wanes over time. And actually within 10 weeks, 10, 12 weeks, 10 to 14 weeks, then it actually decreased to almost zero. Whereas patients who received bivalent vaccine last year will, receive some protection against hospitalization or death a little bit longer, as the graph is showing higher area of area under the curve. And now this year, we are giving the XBB 1.5 monovalent vaccine, and some of you probably have got it already. How does it work with the current circulating strain of EG 5.1? This is an in vitro study, meaning that it's in the test tube. We don't know the clinical data yet because it's not done in a clinical trial setting. We extrapolate the use of XBB 1.5 monovalent vaccine based on the in vitro study. And what they found in the in vitro data is that when you get the serum from patients from individuals who received the XBB 1.5 monovalent vaccine, there is an increased titer of neutralizing antibody at 4 weeks after immunization. And that titer is quite high across the variants against all of these 4 variants, including the EG 5.1. So now, I think the reason why Immunity Canada invited me to give a talk, because I'm the leading investigator for the VISIT study, and I thank all of you who participated in the study or supported the study. So this study includes all 12 centers across Canada. And we have recruited 305 participants so far. We had 189 adults and 116 pediatric participants. Thank you again. So now I'm just going to show you preliminary data. Currently, we are still cleaning our data. There's still a lot for us to do before we can publish our data to the community. But I think we do have something to add. As I mentioned to you, that the Netherlands group only had about 50 patients that they can evaluate the immune response after dose 3. And they only evaluated at 5 weeks after dose 3. But now we have data up to 6 months after dose 4 in our VISIT study. So each dot represents one individual. The blue is from healthy individual, and the red is from immunodeficient patients, the primary immunodeficient patients in our study. So the antibody response is not surprising, similar to other studies in the past, where it shows that patients with immunodeficiency has less antibody response as compared to healthy individuals. This is at dose 2, meaning just before the patients received the dose 2. And these 4 weeks after dose 2, there's an increase in the titer, but still, as compared to healthy, is lower. But, we don't know what the cutoff for protection is, right? Like maybe this level is enough for you to not get infection, period, or not having to go to the hospital for that. Now, what about 4 weeks after dose 3? So if you get dose 3, what happened as compared to healthy control? So the healthy individuals would have very, very high titer after they received 3 doses, whereas our primary immunodeficient patients, there's a range of people. I think there are more than a half, I would say, over here had a very high titer, and then there are certain percentage of people are still having a lower titer. But that then some patients, again, similar to the group, the Netherlands group, some of our patients drop out or did not come back. And we have a certain number of patients who continue to come in at 6 months after dose 4. And it's still showing pretty preserved titer, okay, with regards to antibody response. Now, I do not have the data to show you in terms of subgroup analysis based on the type of primary immunodeficiency, such as X-linked agammaglobulinemia versus CVID versus CID. We do have that data, but it might not be enough for statistical analysis because a lot of our patients are actually CVID patients. And in terms of T-cell response, so similar to other studies as well, that the T-cell response in the immunodeficient patients is lower than the healthy individuals. At 4 weeks after dose 3 is still also lower than healthy individuals. At 6 months after dose 3, also seems like the overall median is lower than the healthy individuals. What about pediatric population? Peds tend to do slightly better than adult immunodeficient patients. Their results are somewhat closer to healthy kids. So immunodeficient kids do very closely to healthy kids in terms of B-cell response as well as T-cell response, which is great to see. And I'll just go over that. And then I only included the data of immunodeficient patients only. I excluded the healthy and just compare adult versus peds. And you will see that although the confidence interval overlapped between those two groups, the concentrations of the dots are more higher in the pediatric population than the adults of almost all time points in terms of antibody response, as well as the T-cell response as well. So I have not done the statistical analysis to show you whether it's statistical significant or not. But overall, the message is peds are probably better than adults. And then people also want to know how many patients had a breakthrough infection and requiring hospitalization. So we only collected the data that require medical attention. We did not include mild COVID infection. So of all the time that we did this, we were doing the study, there were 45 cases reported for medically attended COVID-19 infection. And medically attended, it could be, go to a doctor to try to get antiviral medication. So maybe it's not really having such a significant infection, but because we drill into their minds that when they have an infection, they should call us and let us know so that we can provide the prescription of antiviral agents. So of those, 3 required hospitalization and no deaths at all that we have observed. The first case, actually, I would think that this case is actually not in our study because the patient was enrolled after the patient already had COVID infection, and we did not have their antibody or vaccine response data before the COVID, but I'll just go over it anyway. So this person is a 17-year-old woman who had combined immunodeficiency and already on subcutaneous immunoglobulin. She got COVID infection at 5 months after dose 3. We don't know what was her antibody titer before she got an infection, but after infection, her antibody titer was 6,500 binding antibody units per ml. So that's considered is very high. That was 1 month after infection and 6 months after the dose 3. The interferon gamma spots, which is an indication of T cell response, are also very high, 342 spots at 1 month after infection, and which is corresponding to the 6 months after dose 3. For case number 2, who also was hospitalized, she's a 48-year-old woman with combined common variable immunodeficiency on IVIG. She got infection 2 months after dose 4. And we happen to know her titer before dose 4, and her titer was 629. So basically, having a titer of 629, you still can get infection. But then after that, her titer went up to 2,000 at 6 months after dose 4. And for T cell response, it was 17.5 of interferon gamma spots before she received the dose 4. And it was comparable at 6 months after dose 4. Case number 3, who was hospitalized, she is a 39-year-old woman with combined immunodeficiency, already on subcutaneous immunoglobulin, and she got her infection at 8 months after dose 3. She did not want to get her dose 4. She just waited, and we knew that at 6 months after dose 3, her response to vaccine was not great. Her antibody titer was only 37. And then when she got the infection, we took her blood again at 12 months after dose 3, which is about 4 months after she got the infection. And it was up a little bit more to 131. But despite that, the level is still quite low as compared to healthy individuals who receive vaccine only. Her T cell response was also not great, but after the infection, her T cell response have gone up quite significantly. So all in all, the data is not yet finalized. We're still cooking and doing a lot of data cleaning, and we will share with you next time. Hopefully next time will be in the form of publication. And I just want to reassure everyone that I myself, in my experience, none of my patients ended up in the hospital. And nobody that I know of died of COVID infection for patients with primary immunodeficiency. All right, and I am happy to take any questions.
Thank you. Do you mind? We'll probably save the questions till Bruce is here until after. Hi, Bruce. Until after his presentation, and we'll just have all the questions at the end, if that's okay.
Yes, that's okay.
Awesome. And yeah, welcome to Dr. Ritchie. Do you want to share your screen? That's okay.
Sorry, I'm on mute.
We can hear you.
I'll just share my screen. It's a picture you can see yourself. Oops, wrong screen. It's an intelligence test.
Yeah, that's good. And yeah, it should be perfect. That looks great.
Okay, thank you. Yeah, thanks very much, David. And thanks for inviting me, and congratulations, Juthaporn. Well done.
Thank you.
It's always a worry when you're trying to get promoted. So congratulations.
Yeah, thank you.
Okay, let me just find my screen. So here we go. So first of all, thanks everyone for joining me. The picture of me that David used in the poster was a terrible picture. So I sent him another one, but he doesn't seem to have used it. So that's me. This is my title. It's the Immunity Canada Vaccine Update, November 2023. And just hang on a second. Sorry, and this is just to remind you where we are in Edmonton, little old Edmonton in Canada, and we're on the Treaty 6 territory, traditional gathering place for diverse Indigenous peoples, particularly the Cree, the Blackfoot, the Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and a lot of others. And we just like to acknowledge that and thank them for sharing their land with us. So I'm talking about the vaccines, and I'm going to start with a little, well, my first slide will show what the recent evidence about COVID in Canada shows. So there has been a spike, which you can see here. This goes up to October 21st. The data from October 28th hasn't been posted yet. The website is down here. But if you're trying to figure out what's going on with COVID-19, this is a good website to look at. But as you can see, overall, there's in the vicinity of sort of 15 to 20% positivity, which means not 15 to 20% of the population, but 15 to 20% of the people who have been tested. And this is the current weekly variant breakdown. And as you can see here, the great majority of these are this one here, which is HV.1. And as you go back over the past number of weeks, you can see how this changes over months. So for instance, going back 1 month here, this made up maybe, I don't know, 15%. And now it's probably about a third or so. And so this will continue to change and evolve with time. These are the vaccines, just to remind you. The new vaccines provide coverage for a variant called B.1.1.5, which covers the current things that are circulating. And just to remind you, some of these are RNA vaccines, some of them are viral-based vaccines, like the AstraZeneca vaccines, and then there's protein subunit vaccines. But the ones that have been available in Canada have been mostly the mRNA and the viral vector vaccines, and by far the most used is the mRNA vaccines, and that's the one I've been getting. This is a slide to just remind you of some of the biology of COVID-19 and what it does and how it does it. And so this is a little cartoon of the virus itself. And you can see it's got the knobs on the outside, and these circles on the inside are meant to represent RNA as if it were a double-stranded helix, although this is just single-stranded RNA. But the knobs on the surface are what we call the spike protein, and this shows them binding to a receptor on a cell, which is known as the ACE2 receptor. So that's the interaction by which people get infected. Once this binds to the receptor, the cell internalizes the virus and then releases it into the cytoplasm, where it does its thing, which includes releasing the RNA, which is translated into new virus, which goes through all these processes and then comes out of the cell again. So this background slide is just preparation for the next few slides I'm going to show. So it turns out that you can actually see those viruses under electron microscopy, and this is complicated, but I'm going to go through it briefly with you. This is a picture of a cell with a nucleus. This is a vacuole here, and if you do a close-up on the vacuole, you see this thing here, and the arrows point to little viral particles, and they're enlarged here and elsewhere. And so using even higher resolution electron microscopy, this is what the virus looks like. And it's called a coronavirus because it's got a halo or a corona of what is essentially the spike protein. These slides are from the Rocky Mountain Laboratories in Montana, part of the National Institutes of Allergy and Infectious Diseases, both from the same place. But you know, the key thing is they're called coronaviruses because they've got this corona or halo around them, and that is the spike protein. So just a little background to remind you of what this protein is made out of. It's made out of amino acid building blocks, and this is a picture of those here. The amino acids all have this common sort of structure with a side chain represented by R here. And this is just all the side chains on the 20 amino acids that are essential for mammalian existence, human existence. The key thing is that this backbone here is what makes the chain of amino acids that makes up a protein. So it's interesting that you can have a chain of a long chain of thousands of different amino acids, and they fold into a three-dimensional structure that works most of the time. So that's a background to have a quick look at the spike protein. So this is electron micrograph, so it's electron microscopy to look at the protein itself. And it used to be, to get this sort of resolution of structure, we used to have to purify the protein and try and crystallize it and that sort of thing. But these days, electron microscopy is of such high resolution that you can actually take a picture and then sort out from your knowledge of what the protein chain looks like, what the structure is. And so this gray stuff is the actual picture, and these colored bands are actually what's derived from that. So showing the amino acid chain that underlies this gray stuff. And over on the right here, this is a picture of the spike protein. And again, remember the viral membrane is down here, and this is the spike protein sticking up like an inverted pyramid. And this bit up here is the receptor. And this is a cartoon of it over here. And this is sort of a key structure I want you to pay attention to. So there's this sort of saddle-shaped piece of the spike protein, which is where the ACE2 receptor binds to. And you can see it up here in the same sort of way. This is sort of a saddle-shaped part of the protein. And this is the receptor binding part of the protein. And it's important to know that because that's where we target our vaccines at. So the vaccines are usually made of short pieces or code for short pieces of protein called peptides that represent this particular saddle structure. Because really what you want to do is get your antibodies to bind to this and prevent the virus from binding to your cells and invading them. So this is sort of a common structure. The green stuff here is the receptor binding domain itself. You can see the backbone of the protein. You don't see all the side chains. And the importance of the side chains are that they stick out from this backbone and have, different structures, different charges that are responsible for the actual interaction with the receptor. So this interaction between this green bit here of the spike protein and the blue bit of the receptor. And so I'm going to show a slide with some more details here. This is, again, another picture of the spike protein, and as is this. And what it shows is this green bit, which is the receptor binding part, is quite mobile, and it can be found in a couple of different orientations. So it's got a flexible bit, which allows it to fold down, as you see here. And this picture over here is the original SARS virus, just to show you the similarities of these different coronaviruses. Again, this is another picture of that protein. This here is the receptor binding part here. This is a high resolution picture of that sort of area. And these red bits are the various mutations that happen in the various COVID-19 variants. Now, again, what you're seeing is the backbone. You can't see the side chains sticking up, but it really is the side chains that are interacting with the receptor. So when COVID is mutating and trying to find a way to evade the immune system and things like that, it's changing, in many cases, changing the amino acids in the receptor binding domain, but trying to do it in a way that still allows it to bind to the receptor. And this is just a high resolution picture of the same thing. Now, the thing that COVID does is it evolves. And this is an evolutionary tree of different COVID variants. And the one that's highlighted here is the Omicron variant, B.1.1.5, and it's the one the current vaccine is directed against. And these are the various variants that we've seen. And for instance, EG51 and another one called HV, which I'll show you later, are the ones that are becoming predominant now. And, you know, a question that you would ask is, will a vaccine directed against this guy back here work for these guys down here? And the answer is, we hope so. And in most cases, the answer is yes. Because these guys here have all evolved from this one, and because when they evolve, they're evolving in a way that allows them to change enough to try and evade the immune system, but not so much that they can't bind to the receptor. So there is a lot of structural similarity between all these variants here, XBB 1.1 and 1.5, and its offspring. There are changes, but the structure is very similar to this one back here. And so the vaccine directed against this variant is thought to be still active against those other ones. So this is an evolutionary tree similar to what you see in human and other evolution, but this is just for the virus. And there's many more complicated ones that go back further to try and look at the evolution of the original COVID virus and its relation to the SARS virus and other coronaviruses. But this is the bit that's sort of important here. And again, Omicron, B.1.1.5 is the one that the current vaccine that was released in October is directed against. And again, this picture shows what the common variants are. HV1 and EG5 are the ones that are most prevalent now. And you can see that these have changed significantly over the last month, and we expect that they will continue to evolve. So what about the vaccines themselves? This is a picture from CBC News talking about the XBB.1.5 variant, subvariant, which again is a variant of the Omicron variant, and it is thought that it will be covered, and early evidence suggests that it does give coverage for the current variants. So one of the questions you might have is, what about SARS-CoV, or what about the COVID-19 infection itself, and how does that interact with immunodeficiency? And so there's a couple of interesting papers that I just thought I'd show you here. And this one speaks to what happens if you've had COVID-19. And this is looking at people without immunodeficiency and suggests that in many cases, the COVID-19 itself gives you immunodeficiency. So one of the concerns about this is, if you're already immunodeficient, will COVID-19 make you even more immunodeficient? And it's not known for certain, but there is concern that that is exactly what happens. The other question is, what about long COVID? Are people more likely to get long COVID if they're immunocompromised, if they have immunodeficiency, either primary or secondary? And the answer is, it's complicated. And so this is a paper that was published in the European Journal of Pediatrics last year. And their conclusions were that long COVID is characterized by persistent symptoms following COVID-19, and it's due to an effect on various tissues, particularly the brain and other things. And the course among children with or without immunodeficiency shows that the prevalence and severity of long COVID is similar in both groups, except that immunocompromised kids seem to suffer from long COVID, but the prevalence is lower than in normal immunocompetent children. So that was a surprise. You might think that kids with immunodeficiency get worse COVID and are predisposed to more long COVID, but that was not the case. The potential causes of less frequent and milder long COVID in this group may be a milder course of COVID-19. There's all kinds of reasons that might happen, such as treatment of their COVID-19 because they're already immunocompromised, but we don't know for sure. When we look at adults, there's this paper published last year again, looking at a systematic review, I should say, in which they looked at all the publications to try and figure out if you have immunodeficiency, what are the outcomes? And these are the results here. They looked at 69 articles and 459 people who were covered by these 68 articles, all with primary immunodeficiency and infection with COVID. Overall, the case fatality rate, the death rate was 9%. Hospital rate was about 49%, about half those patients were admitted, and O2 supplementation was about 30%. They suggest that overall, people with primary immunodeficiency, when infected, tested positive and showed symptoms for similar lengths of time as the general population. That primary immunodeficiency patients in these papers had some immune response to the COVID vaccine, but it varied depending on the type of primary immunodeficiency you had, as Juthaporn commented, and other factors, other concomitant health disorders, particularly diabetes and things like that. The vaccine was effective in most of these primary immunodeficiency patients, but gave less protection against hospitalization. So in this series of papers, people seem to have more severe disease and require hospitalization, although we don't know. We don't know if these people were hospitalized more frequently because people suspected they might get sicker or because they were actually sicker. There are some strategies, and I think these are really important, such as pre-exposure prophylaxis. And for a year or so, we used to give out this antibody preparation called Evusheld. It was a prolonged half-life monoclonal antibody, actually 2 different monoclonal antibodies, but it doesn't give coverage for the Omicron variants that are around now. So we've stopped giving it. AstraZeneca, who produced this, have just finished a trial for a new version, which we hope will give Omicron coverage. And then there's a whole bunch of drugs, which I'll talk about briefly. One of the things, and Juthaporn alluded to this, is people with X-linked agammaglobulinemia, which is a more severe antibody deficiency, they seem to have more prolonged infections. And by that, I mean they don't seem to get better more quickly. And so it's important in all the immunodeficiency patients, but particularly the X-linked ones, to think about getting treatment with a medication or whatever antibodies are available at the time if they get infected with COVID-19. And here are just some of the questions that were sent to the website that David provided me. And I thought in the interest of efficiency, I'd just zip through these most quickly with my opinion. I've heard that the current variant XBB1 is especially dangerous for immunodeficiency patients. Will the current vaccines available protect against XBB1? And the early evidence is that they do give protection. We're not sure if it's as good as for the other variants, but it looks like there is protection. Does having primary immunodeficiency make someone more susceptible to COVID, long COVID? Are there any statistics comparing the similar percentage or comparing the percentages of patients against the general population? And I showed you data on that, in that they seem similar. You know, patients may not generate quite the same immune response, although they seem to get some immune response. and they may not respond to vaccines quite as well, but they seem to get a similar risk of COVID-19 and similar degree of severity. And how effective are the vaccines against the current strains? I think they're pretty good, but results are still coming in. So the vaccine's only been around for a short period, and as we mentioned, the variants are evolving at an ongoing rate. So we'll see what the protection is over the next few months. Will there be continued boosters? And I think there will be boosters forever, or at least until we can find a way to eradicate COVID-19. And remember, we have eradicated some viruses like smallpox in the past. So it's not impossible to think that we might be able to get rid of COVID-19. But at the present time, it looks like it's going to be like the flu, and that we have to get new vaccinations at least every year. What's your advice for dealing with people who ask why we wear masks in public, like at the supermarket? My advice is down here: ignore people who comment. And I said, I smile at them, which of course they can't see because I've got a mask on, or MYOB, mind your own business. But you should wear a mask. You shouldn't worry about it. I'm not immunocompromised, but I wear a mask, particularly when I'm on airplanes, and it's just something I do, and people need to get used to that. Do we need vaccines, or are the COVID-19 antibodies that we get in the plasma supply effective against the current variants? And the answer is you definitely need the vaccines. The antibodies that are in the plasma supply are from at least 18 months ago. So they're against previous variants. So it's unlikely or less likely that they're going to give you good coverage for the current variants. So you must get the vaccine. I can't stress that enough. What kind of COVID-19 treatments or therapy are available for people with primary immunodeficiency? And the current oral one is Paxlovid. There's a bunch of others that I'll show you on the next slide. I have a fear of catching COVID-19. Do you have any statistics about the number of people dying? And as I showed on the previous slide, it's about 9%. So it's still a serious thing, and you should try and avoid it if you can. As the disease goes on, it seems to be something we're tolerating a little bit better all the time because of the antibodies we've developed either to vaccination or previous exposure. And how are the long-term symptoms of COVID-19 diagnosed for people who have a PI? Often people are a little worse acutely, but the long-term results are similar. So the number of patients getting long COVID seem to be similar, and the severity seems to be similar. And what outcomes have been seen in the PI community for long COVID? As I mentioned, they seem to have similar results to those without immunodeficiency. But I think the critical thing is that long COVID seems to last a long time, and there are a variety of pathologic problems that are happening. I'm just going to finish off quickly.
Dr. Ritchie, do you mind going back to that slide just for a second?
Yes.
And I just wanted to ask Dr. Cowan, is there anything that you'd like to comment on with any of those responses at all?
Oh, I agree with Bruce. Yes, definitely need vaccination. Antiviral, Paxlovid, remdesivir can also be used. Yeah, no, all is good.
Awesome, thank you. And Bruce, just to let you know, we'll try and wrap your presentation up soon so we can get more into Q&A session as well.
Yeah, last slide here. This is the currently approved treatments in Canada. Evusheld doesn't cover the current variants. Paxlovid is a couple of antivirals. There's also remdesivir, as Juthaporn mentioned. And then there's a host of monoclonal antibodies. This one, tocilizumab, is actually against interleukin-6. It's an interleukin-6 inhibitor. And so it's not against the variant itself, but what it does is it prevents you from having severe immune reaction to the virus. These others are against the virus itself, and they may or may not cover current variants. And so if you have symptoms of COVID, if you test positive for COVID, then you must dial the 118 or 119 number, whatever it is, and see if you qualify, because I think it's important that you do get treatment in view of your immunodeficiency, and whoever you get on the line will tell you what the best treatment is for the current variants. And I'll finish there and ask if there's any other questions.
Awesome. Thank you so much to both of you. I'm just going to stop sharing your screen real quick, and then I'm just going to pull up my one. I know that Dr. Cowan, you've been answering some of the questions directly in the Q&A. We had some submitted as well. First question to both of you. This person said that I'm 74 years of age, have PID and an autoimmune disorder with SCID infusions weekly. I've had COVID twice. I do not do well with vaccines and wonder why. I was so sick from my second vaccination. For 3.5 weeks, I suffered with a massive migraine headache and symptoms of COVID, then another 2 weeks of healing, so very frightened to have another. Why do vaccines, be it flu or any other, affect one so intensely, and do others have this type of response?
I can start to answer some of the adverse event following immunization piece. We don't know for certain why some people have adverse event and some people don't. Like we don't know why young men would have more myocarditis, pericarditis than other patient populations. And I noticed myself as well, like just give my personal example with the COVID vaccine, I get more reaction than influenza vaccine. And certain vaccine like Shingrix, many people will tell you so that it's so strong. You can get very high fevers because of the high reactogenicity with the added adjuvant in that vaccine. So mRNA, for some reason, it induces a very strong immune response, and therefore some people can have some inflammatory response towards it. Yes, you can be scared. And the other thing is you can be scared of getting the vaccine. I was scared too after my second dose. I was down with swelling of my ankle joint, and even though I did not have any inflammatory arthritis before. But the third dose, the fourth dose, the fifth dose was okay. I did not have any reactions again. And we do have data in our special immunization clinic network across the country, seeing patients with adverse event following immunization. Usually, the events do not happen again following revaccination for whatever reason.
Yeah, and I just jump in and say that the reactions seem to be quite variable from person to person and from vaccine to vaccine. So, just because you had a problem with a previous vaccine doesn't mean you're going to have a problem with the other vaccine. However, if you've had, an adverse event with the Moderna vaccine, it might be useful to try the Pfizer one next time, or the AstraZeneca. That's probably the best I can suggest. I'm an old guy. I'm 69 years old next week. And so I'm a big fan of vaccinations because I think it's important to stimulate your immune system. And that helps to protect you against other things like cancers and stuff. So I've had everything. I've had the RSV vaccine, the combined high dose flu vaccine, and COVID vaccine, and Shingrix, and hope to get every vaccine I qualify for.
Awesome. Thank you both. And the next question is, with the reduction of monitoring data regarding COVID, I'm wondering what metrics you suggest we use for navigating risk and exposure. What information should we be looking at, and where can we access them? It is hard to know how to balance life, isolation, and risk without having much data.
Well, in the one slide I showed, it gives you the data from about a week or so ago. And I think the data from the October 28th will be out pretty quickly, like this weekend. And, you know, so the monitoring hasn't really gone down. I think we're still doing monitoring and still getting results and still publishing them. I think people are getting tested less frequently. But I think there's pretty good data about what's happening. And as we've seen, there's been a spike over the last month or two. So there is good data. And, you know, I think when you go out in public, if you're immunocompromised, you should wear a mask and take care.
Awesome, thank you. And Dr. Cowan, I know you've been busy answering questions in the Q&A. Is there anything that you wanted to comment on with some of those questions, or?
So the questions that come in through Q&A, do other attendees see as well, or it's just direct to us and then the answer doesn't go out to everyone to see?
The answer, I think, goes to everyone in the Q&A.
Yeah, then no, I have nothing to add unless anyone has any additional question.
Brilliant. No, I think we've got to a lot of the questions that relate to this, and yeah, just to kind of wrap things up, I just wanted to say again, thank you both so much for taking the time and to be with us today, and a big thank you again to Moderna for helping us put this on. And if we didn't get to your questions or you have more, feel free to submit them to us. You can reach us at info@immunitycanada.org, and always look at our website to see what other events are happening. We have some pediatric immunologists talking about having a Q&A session at the end of the month, and we've started patient and family community connect groups across Canada. And Fiona's been busy planning a lot more volunteer opportunities for everyone to get more involved. And yeah, a huge thank you again for attending, and yeah, feel free to reach out to us. Awesome. Well, thank you both, and we'll see you again soon.
Okay, bye-bye.
Bye now. Bye.